Welcome back to Rad on Smart Review, the RadBio edition.
Hey, everyone.
Today we're going to tackle a really core concept, homologous recombination.
Yeah, HR. It's absolutely fundamental for us in radiation oncology. We really need to understand this pathway.
Right, how cells fix those really dangerous DNA double strand breaks, the DSBs.
Exactly. And HR is the high fidelity way they do it. It's crucial for understanding radiation effects and, even cancer therapy.
So when radiation hits DNA and causes a double strand break, the cell has choices, right? There's NHEJ.
Right, non-homologous end joining. That's the quick and dirty method, like just sticking the broken ends back together.
Yeah.
Fast, but yeah, it can make mistakes, lose information.
And HR is different?
Totally different.
Yeah.
Think of HR as the meticulous expert, the restoration specialist.
Right.
It doesn't just patch things up. It aims for a perfect, error-free repair.
Okay, a meticulous restoration expert. I like that. How does it manage that? Where does it get the information to be so perfect?
That's the key. It uses a template, an undamaged, identical DNA sequence.
And that template is?
The sister chromatid, the exact copy that's made during DNA replication.
Ah, okay. So if it needs the sister chromatid, that must limit when HR can actually happen, right? During the cell cycle.
Precisely. You only have the sister chromatid readily available after the DNA has been duplicated.
So we're talking late S phase and G2 phase.
Exactly, late S and G2. HR isn't really a major player in G1 because, well, the template just isn't there. That's more NHEJ territory.
Got it. Late S and G2, super important point for boards and just understanding this. Okay, so how does this meticulous repair actually unfold? What are the steps?
It's a really elegant multi-step process. It starts, as you'd expect, with recognizing the break.
Okay, first responders, who are they?
That's primarily the MRN complex.
Yeah.
MRE11, RAD50, and BS1. They're like the initial sensors, and they work with ATM kinase.
ATM, the big signaling kinase.
Right. ATM gets activated and starts sending signals, basically telling the cell, hey, we've got a serious problem here.
Alarm bells ringing. Okay, break detected, signal sent. Then what?
Then comes a really critical step that defines HR end resection.
Resection, like trimming the ends.
Kind of, yeah. But specifically, nucleuses chew back the 5' ended strands at the break site.
Okay, chewing back the 5' ends.
Exactly. This creates these long single-stranded DNA tails with a 3' end. These are called 3' overhangs.
And who does this chewing?
Key players include the MRE11 nucleus activity within the mRNA complex itself, plus proteins like CTIP, XO1, and DNA2.
Right. And this resection step is important because
it essentially commits the break to the HR pathway.
Okay.
It creates the substrate needed for the next steps. It's also kind of a decision point. In G1, a protein called 53BP1 actually blocks extensive resection, favoring NHEJ.
Ah, so 53BP1 is like a gatekeeper pushing things towards NHEJ in G1, but in SG2, resection proceeds favoring HR. Makes sense.
Exactly. So now you've got these long, fragile, single-stranded DNA overhangs. They need protection.
Okay, what protects them?
That's where RPA comes in, replication protein A. It's a single-strand binding protein, and it just rapidly coats those ssDNA overhangs.
Like putting protective insulation on a wire.
Yeah, that's a good way to think about it. Stabilizes them, prevents them from getting degraded or forming weird structures.
Okay, RPA is on. What's the next big step? This sounds like it's leading up to the actual repair part.
It is. This next step is formation of the RAD51 filament. This is absolutely central to HR.
RAD51. I know that name. That's the recombinase, right?
The core recombinase, exactly. But it needs help getting onto that RPA-coded ssDNA. RPA has to come off first.
And who helps with that? I feel like this is where BRCA comes in.
You got it. Specifically BRCA2. Think of BRCA2 as a chaperone or maybe a foreman. Its job is crucial. It helps remove RPA and then loads multiple RAD51 proteins onto that ssDNA overhang.
So BRCA2 boots RPA off and recruits RAD51 workers onto the job site.
Precisely. This forms the RAD51 nucleoprotein filament. It's essentially RAD51 coating that single strand. RAD52 also plays a role here, assisting RAD51 loading and protecting the DNA ends too.
Okay, so now we have this RAD51 filament sitting on the processed DNA end. What can it do?
This filament has an amazing capability. Homology search. It can actually search through the genome.
Searching the whole genome.
So searching locally for its match, its identical sequence on the sister chromatid.
Wow. And when it finds it?
Strand invasion. The RAD51-coded ssDNA overhang literally invades the double helix of the undamaged sister chromatid. It displaces one of the sister strands and pairs up with its complementary sequence.
It invades the sister DNA? Okay. That forms a D loop, is that right?
Exactly, a displacement loop or D loop. You've got this temporary three-stranded structure.
Okay, that sounds complex but necessary. What happens once that D loop is formed?
Now the actual repair synthesis can start. The invading 3' end acts as a primer.
A primer for DNA synthesis.
Yep. DNA polymerases, often pol delta, just like in replication, are recruited. They start synthesizing new DNA using the undamaged sister chromatid strand as the perfect template.
Ah, so that's how it copies the lost information accurately, using the sister as the blueprint.
Exactly right. High-fidelity repair in action.
Okay, since this is happening, are we done?
Almost. This whole process, invasion, synthesis, it creates these complex branched DNA structures. The classic one is called a holiday junction.
Holiday junctions, right? Those X-shaped structures where the chromosomes are linked.
These junctions connect the original damaged molecule and the template sister chromatid. They need to be resolved, basically cut apart neatly, to separate the two DNA molecules again.
And how does that happen? Special enzymes?
Yep. Specialized enzymes called resolvases cut the holiday junctions. This resolution can happen in a couple of ways, leading either to a non-crossover outcome, just simple repair, or a crossover outcome, where the DNA flanking the original break site gets exchanged between the sister chromatids.
Okay, resolution happens. Then the final touch.
Just sealing any remaining nicks in the DNA backbone. DNA ligases like LIG1 come in and do the final ligation step. And voila, you have two intact DNA molecules perfectly repaired.
That's quite the journey. From break detection all the way to ligation. Really highlights how complex but elegant this is.
It really is.
We mentioned BRCA2 loading RAD51. What about BRCA1? It's always talked about alongside BRCA2. Where does it fit in?
Great question. BRCA1 is also absolutely critical, but its role is a bit different. It acts more upstream and as a coordinator. Think of it like the project manager or maybe the site supervisor.
Okay, BRCA1, the manager, BRCA2, the foreman. I can work with that. What does the manager do?
Several key things. First, BRCA1 gets to the break site very quickly and is involved in checkpoint signaling. It talks to ATM and ATR to help pause the cell cycle, giving the cell time to fix the damage.
Right, putting the brakes on division. Makes sense.
Second, it actively promotes that end resection step we talked about. It helps counteract proteins like 5,3-BP1 that would otherwise block resection and push towards NHEJ. So BRCA1 helps choose the HR path, especially in SNG2.
So it helps commit the cell to high-fidelity repair.
Exactly. And third, it acts as a scaffold. It interacts with a whole bunch of other repair proteins, including the MRN complex, CTAP, and it even influences BRCA2 and RAD51 function. It helps bring everyone together.
Wow, okay. So BRCA1 is really central to orchestrating the whole response.
Definitely.
Yeah.
Both BRCA1 and BRCA2 are indispensable for efficient HR.
Which leads us to what happens when they aren't working. When you have mutations in BRCA1 or BRCA2.
That leads to homologous recombination deficiency, or HRD. The cell's ability to perform this high-fidelity repair is severely compromised.
The meticulous expert is off sick.
Pretty much. And the cell becomes much more reliant on the error-prone pathways like NHEJ.
And relying on the error-prone pathway means?
Genomic instability. Lots of mutations, deletions, chromosome rearrangements accumulating over time. The genome gets messy. And this instability is a major driver of cancer development.
Which is why inherited BRCA mutations lead to hereditary breast and ovarian cancer syndrome, HBOC.
Precisely. That dramatically increased risk is a direct consequence of impaired HR and the resulting genomic instability.
So understanding HRD isn't just about basic science. It has huge clinical implications.
Absolutely massive. First, as we said, cancer predisposition syndromes like HBOC. Second, HRD tumors often have like genomic scars or patterns that we can sometimes detect as biomarkers.
Okay. And therapeutically, how does knowing a tumor is HR deficient help us treat it?
This is where it gets really powerful. Because these HR deficient cells have a fundamental weakness in DNA repair, they can be exquisitely sensitive to certain types of treatments.
Treatments that cause the kind of damage HR normally fixes.
Exactly. Think about platinum-based chemotherapy, cisplatin, carboplatin.
Right. They cause crosslinks in DNA.
Yes. And when the cell tries to replicate DNA with those crosslinks, the replication fork stalls. And this often leads to double-strand breaks.
Ah, so they indirectly cause DSBs.
Right. Normal cells can handle these DSBs using HR. But HR deficient cells, they struggle. They can't repair that damage effectively, making them much more sensitive to being killed by platinum agents.
Okay, so HRD predicts sensitivity to platinum chemo. What else? The big one everyone talks about, PREB inhibitors.
PRP inhibitors. This is the classic example of synthetic lethality in action.
Synthetic lethality. Explain that concept again.
Okay, so PRP enzymes are key players in repairing single-strand DNA breaks, a different type of damage. If you inhibit PRP using a PRP inhibitor,
Those single-strand breaks don't get fixed.
right, and if they persist, especially when the cell tries to replicate its DNA, they can get converted into double-strand breaks.
Okay, so inhibiting PRP leads to more DSBs.
Yes. Now, a normal cell with functional HR sees these DSBs and says, okay, no problem, I'll use HR to fix these.
But the HR deficient cell,
The HR deficient cell, like one with a BRCA mutation, can't fix those DSBs efficiently using HR because its HR pathway is broken. And it also can't fix the initial single-strand breaks because PRP is inhibited.
so it's hit from two sides. It can't fix single-strand breaks, and it can't fix the resulting double-strand breaks.
Exactly. The combination of the two defects, the inherent HR deficiency and the drug-induced PIRP inhibition, becomes lethal to the cancer cell. That's synthetic lethality. One defect alone is tolerable, but the two together are fatal.
A really elegant therapeutic strategy targeting a specific vulnerability.
It really is. It's been a game changer for patients with BRCA-mutated cancers.
What about radiotherapy? Radiation causes DSBs directly. Should HR-deficient tumors be more sensitive to radiation too?
Theoretically, yes. It makes perfect sense. If you can't efficiently repair the main lethal lesion caused by radiation, the DSB, you should be more radiosensitive.
And is that what we see clinically?
There's definitely preclinical data supporting it and some clinical hints as well. But, the clinical picture is often complex. Tumors have lots of other mutations. The microenvironment plays a role. It's not always as clear-cut as with PRP inhibitors. But it's a very active area of research trying to understand if and how we can leverage HRD to predict or enhance radiation response.
Okay, lots to think about there. Let's crystallize some of this with our clinical pearls. What's the first takeaway?
Clinical pearl number 1. Remember, homologous recombination is your high-fidelity, error-free repair pathway for DNA double-strand breaks. Key things. It needs the sister chromatid as a template, and it's mainly active in late S and G2. High-fidelity sister SG2.
Nice. HR, high-fidelity sister, SG2, pearl number 2.
Clinical pearl number 2. Don't forget BRCA2's direct role. BRCA2 protein is essential for loading RAD51 onto that single-stranded DNA. Think BRCA2 equals RAD51 loader.
BRCA2 loads RAD51. Got it. And the third pearl.
Clinical pearl number 3. Defects in HR, especially BRCA1 or 2 mutations, cause genomic instability but also create therapeutic vulnerabilities. Specifically, think sensitivity to platinum chemo and crucially synthetic lethality with PRP inhibitors. HR deficiency, therapeutic target.
HRD is a target. Great summary, pearls. So how does this knowledge impact actual clinical decision-making day to day?
Well, the most direct impact is testing for BRCA1 and BRCA2 mutations, and increasingly other HRG mutations or HRD signatures.
Right, in cancers like ovarian, breast, prostate, pancreatic.
Exactly. If those mutations or an HRD signature are present, it opens the door for using PREP inhibitors, often significantly improving outcomes.
And it influences chemo choices too.
Yeah, it can definitely weigh into the decision about using platinum agents. A known HR defect might make you lean more towards platinum if it's an appropriate option for that cancer type.
And the radiation link we talked about?
Still mostly in the research realm, but definitely informing trial designs. People are looking at whether HRD status predicts benefit from specific radiation doses or fractionation schedules, or maybe combining radiation with drugs like PRP inhibitors to exploit that potential radiosensitivity. It's an evolving area.
Okay, makes sense. Time for a quick board blitz. Let's see if we've got these key concepts down.
Let's do it. Fire away.
Question 1. Homologous recombination HR repair of double-strand breaks primarily occurs during which phases of the cell cycle? Is it A, G1 phase only, B, M phase only, C, G1 and S phases, or D, late S and G2 phases?
Okay, HR needs that template, the sister chromatid. That's
Yep.
only around after replication, so it has to be D, late S and G2 phases.
That's correct. Rationale. HR requires the sister chromatid available in late S and G2. Question 2. Which tumor suppressor protein plays a critical role in loading the RAD51 recombinase onto single-stranded DNA during homologous recombination? A, T53, B, ATM, C, BRCA1, D, BRCA2.
Right, the RAD51 loader.
The format.
That was D, BRCA2.
Exactly. Rationale. BRCA2 facilitates RAD51 filament formation on ssDNA. Okay, question 3. Compared to non-homologous end joining NHEJ, homologous recombination HR is characterized by A, lower fidelity and activity throughout the cell cycle, B, higher fidelity but restricted activity mainly to G1 phase, C, lower fidelity but restricted activity mainly to SG2 phases, or D, higher fidelity and restricted activity mainly to SG2 phases.
Okay, HR is the meticulous one, so higher fidelity. And we just established is mainly late SG2, so that points to D.
Perfect. D, higher fidelity and restricted activity mainly to SG2 phases. Final one. PIRP inhibitors show particular efficacy against tumors with BRCA1 or BRCA2 mutations due to the principle of A, oncogene addiction, B, bystander effect, C, synthetic lethality, or D, horizontal gene transfer.
The PRP inhibitor mechanism in BRCA mutated cells. That's the classic example of C, synthetic lethality, blocking two repair pathways.
You nailed it, rationale. Blocking SSB repair, PARP, in HR-deficient cells leads to lethal accumulation of DSBs. Great job.
Good questions. Definitely board-relevant stuff.
Absolutely. Now, moving beyond the basics, what are some of the current, maybe controversial or advancing areas around HR? What are people debating or actively working on?
Well, a big one is definitely how we actually define and measure HR deficiency. It's not always as simple as just finding a BRCA1 or BRCA2 mutation.
Right. Other genes are involved, PALB2, ATM, Cheeky2.
Exactly. And sometimes the gene looks fine, but the pathway isn't working right for other reasons, maybe epigenetic silencing or protein-level issues. So the big challenge is, how do we reliably identify all patients whose tumors are functionally HR deficient?
And determine if they'll respond to PRP inhibitors or platinum like the BRCA mutated ones do.
Precisely. There's a lot of work on developing better biomarkers, looking at genomic scars or signatures of HRD, like specific patterns of mutations or chromosome changes across the whole genome. But standardizing these, improving their clinical utility across different cancers, is still ongoing. It's complex.
So moving beyond single genes to functional pathway assessment, what about other advances? New drug targets?
Yeah, definitely. Researchers are looking at other proteins in the HR pathway as potential drug targets, maybe inhibiting other key players like RAD51 itself, or proteins involved in resection like MEM11 or CTIP.
To make cancer cells even more sensitive to damage.
Exactly, or maybe to overcome resistance to existing therapies. On the flip side, there's also interest in finding ways to boost HR activity, but specifically in normal tissues.
To protect them.
Right, to potentially reduce the side effects of chemo or radiation on healthy cells by making them better at repairing the damage. That's more challenging, but it's an interesting idea. And of course, refining those HRD biomarkers is a huge area of advancement.
Fascinating. A lot's happening beyond BRCA and PRP inhibitors. Okay, let's wrap this up with a quick summary of our deep dive into homologous recombination.
Sounds good. So we establish HR is the crucial error-free pathway for fixing DNA double-strand breaks. Its defining features are using the sister chromatid as a template and being most active in the late S and G2 phases.
We walk through the key steps. Break recognition by mRN and ATM, end resection creating 3' overhangs, RPA coding.
Then the critical step of RAD51 filament formation, heavily dependent on BRCA2 acting as the loader, followed by homology search, strand invasion forming a D-loop.
DNA synthesis using the sister template, resolution of intermediates like holiday junctions, and finally, ligation.
And we highlighted the roles of BRCA1 as the upstream coordinator and BRCA2 as the direct RAD51 loader.
Critically, we discussed how defects in this pathway, especially BRCA12 mutations, lead to genomic instability and cancer predisposition, like in HBOC.
But also how this deficiency creates therapeutic opportunities, increased sensitivity to platinum chemotherapy, and, very importantly, synthetic lethality with PRP inhibitors.
Making HR status a key factor in modern oncology treatment decisions.
Absolutely. Understanding HR is just vital for any radiation oncologist today.
Well, this has been incredibly informative. Thanks for breaking down such a complex topic.
My pleasure. It's fundamental biology with direct clinical relevance.
For everyone listening, make sure to solidify this knowledge. Maybe check out practice questions over at redonsysmartreview.com and definitely subscribe so you don't miss our next deep dive.
Keep learning.
Thanks for tuning in.